Enterobacteriaceae gram-negative bacilli, all of them are oxidase negative, catalase positive, and reduce nitrate to nitrite. All ferment glucose. They are divided into two groups. One is lactose fermenter and other is non-lactose fermenter. In lactose fermenter, we have E. coli, Klebsiella, Citrobacter, and Enterobacter. E. coli and Citrobacter are methyl red positive, and as the name indicates, Citrobacter is citrate positive and E. coli is citrate negative. Further, the methyl red negative organisms are Klebsiella and Enterobacter, and both are being divided on the basis of motility. Klebsiella is non-motile and Enterobacter is motile. Further, Enterobacter species are divided on the basis of lysine. Lysine's test is positive for the erogen and Sakazaki, while lysine test is negative for Enterococcus colaceae. For Klebsiella pneumoniae, which motility is negative, further divided on the basis of indole. If indole is positive, then it's Klebsiella oxytoca, and if indole is negative, then it is Klebsiella pneumoniae. The one of the largest group in this chart is NLF, non-lactose fermenter, which is Salmonella, Shigella, Yersinia, Margonella, Margoni, and Serratia, Advarsella, Proteus, and Providentia. They are also differentiated on the basis of methyl red. Methyl red negative species Serratia, while all other species are methyl red positive. Further, the another test is motility, which distinguishes the Shigella from the whole group. Shigella is the non-motile organism, and mannitol is further used for the division of Shigella. In mannitol negative is Shigella dysentery, while all the three species, Phylloxera, Bodi, and Suni, are mannitol positive. ONPG is a very useful test because it is positive for Shigella soni and negative for Shigella flexneri and Bodi. Further, the motile organism like Proteus, Providentia, Salmonella, Yersinia, Advarsella, Margoni is further subdivided on the basis of PAD test. PAD is phenylalanine diaminase test, in which the agar slant is inoculated at 37 degrees centigrade for 12 hours, and then ferric chloride 10%, 4 or 5 drops are added, and we check for the color change. If the color is changed to dark green color, then the Margonella, Margoni, Proteus, and Providentia is suspected, and these organisms are further distinguished on the basis of H2S production, in which the Proteus is the major only organism in PAD positive organism, that is H2S producer. Proteus is further distinguished on the basis of indole, where the Proteus mirabilis is negative and Proteus vulgaris is positive, and the organism is also can be differentiated on the basis of citrate, in which mirabilis, which is indole negative, is citrate positive, while vulgaris, which is indole positive, is citrate negative. And the other we can say the other differentiation in the Proteus species is a citrate. Other than citrate is a TSI, in which the Proteus mirabilis show the alkaline by acid plus weak H2S and causing major cause of UTI 90%, and the vulgaris, which is the major cause of the wound infection, and that produce acid by acid and H2S in a large amount. Further, in this chart we will go to the PAD negative organism, which is Salmonella, Advarsella, and Yersinia, which is in which we can differentiate on the basis of H2S production. Advarsella and Salmonella is positive for H2S, and Yersinia is negative for H2S production. Yersinia enterocolitica is positive for ODC, and pertussis and pseudotuberculosis are negative for ODC. Further, pertussis and pseudotuberculosis can be differentiated on the basis of motility at 25 degrees centigrade, where the pseudotuberculosis is positive for motility and pertussis is negative. Further, if we see the differentiation between the Advarsella and Salmonella, which is H2S product, which can produce the H2S. Indole is the only test which differentiate between Advarsella tarda and Salmonella. Indole positive is tarda and negative is Salmonella, but Salmonella is also citrate positive, while tarda is also citrate negative. Both produce alkaline by acid plus H2S, and that's it. Curve gram-negative bacilli. Curve gram-negative bacilli are few organisms that are medically important. Number one is Campylobacter jejuni that have the corkscrew motility and mostly common cause of the bacterial diarrhea. The organism is oxidase, catalase and evaporate positive, and also have ability to grow on 42 degrees centigrade. And regarding susceptibility, it is sensitive to nalidixic acid and resistant to cephalothene. Campylobacter fetus is similar to the jejuni, but they do not produce the evaporate and also do not have the ability to grow on 42 degrees centigrade, and also opposite susceptibility pattern with the nalidixic acid and cephalothene to Campylobacter jejuni, because it is resistant to nalidixic acid and sensitive to cephalothene. And H. pylori is quite different from both of them because it is also oxidase and catalase positive, but the organism have ability to produce the urease, which urease enzyme give this organism stability to survive the harsh environment of stomach, and its susceptibility pattern is quite similar to the Campylobacter fetus, which is nalidixic acid resistant and cephalothene sensitive. The organism do not grow on 42 degrees centigrade and also do not hydrolyze the evaporate. Another organism in this group is Vibro Cholera, which is a major cause of rice water stool and extreme dehydration because of its toxins. The organism is oxidase positive, and the unique ability is sucrose utilization, so will produce the yellow colonies on TCBS, and also the positive string test is also one of the major you can say prognostic marker in early diagnosis. Vibrio villanificus and parahemolyticus is found in the salt water, and the organism is unable to ferment the sucrose, so they will produce the green colonies on TCBS. And also, as you know, it is a salt water organism, so need the sodium, and also we can say it's halophilic. Aeromonas and Pseudomonas both are oxidase positive, and these are the only organism in the Enterobacteriaceae group that are oxidase positive organism, but because all of other Enterobacteriaceae are oxidase negative. Aeromonas is freshwater and saltwater both, and again the Pleisomonas schedulodes are freshwater and seafood poisoning. Basically, both of these organisms are the major cause of disease in cold-blooded animals like fishes and other consumable product for seafood in the humans. And Aeromonas is also cause the skin infection tsunami, and it can be differentiated because of the biologically positive. And this organism have three major groups that are mostly found in the human infections. Pleisomonas schedulode, it's quite similar to the schedulosis disease, and that's like the bloody stool, so that's why. And the toxins are quite similar to the schedulode, and that's why it's Pleisomonas schedulodes. Further, we will discuss these two organisms in detail in other videos. Now we will go to the gram-positive cocci. In gram-positive cocci, we are differentiated on the basis of catalase test. Catalase positive organism are Staphylococcus aureus and Micrococcus, while negative is Streptococcus family. First, we will discuss the Staphylococcus and Micrococcus. The other distinguishing test is coagulase, which is positive for Staphylococcus aureus and intermediate Staphylococcus aureus. Intermediates are organisms that have salt tolerant, beta hemolytic, exfoliative enterotoxins which cause the scarlet skin syndrome, and also the organism hyaluronidase production, and also the organism is trypanosome cause TSS, toxic shock syndrome, and 50% us we carry this organism on nares and skin, and the organism is a beta lactamase inactivator, especially in the case of MRSA, so treat with the vancomycin. Further, the coagulase negative organisms are staph and micrococcus. We done the oxidase or microdase or bisitracine or liphostaphine, which is positive and sensitive for the micrococcus and negative for the coagulase negative staphylococcus. In coagulase negative staphylococcus, we use the novobicin. Novobicin sensitive is staph epidermidis. The zone of inhibition is more than or equal to 16 mm. Another thing is the hospital acquired opportunistic pathogen get into the surgical wounds and cathedral to cause the UTI, possesses the glycocalyx, capsule slime layer, and the biofilm producer bacteria is staph epidermidis, which is normally found in the skin as a normal flora. And now bicin resistant or staphylococcus saprophyticus urinary tract infection cause UTI in the sexually active females. Now we will go for the streptococcus family. Streptococcus family, we have beta hemolytics and alpha hemolytics. First we will disclose alpha hemolytic. In alpha hemolytic we have Optochin's disc, which is sensitive to the pneumonia and the susceptibility zone is more than 14 mm. This is lancet shape in the pairs and glistening with doom colonies, bile solubility positive, quelling reaction positive, capsule swelling, and required of carbon dioxide, brain heart infusion agar, tryptocase soya broth, chocolate agar, polysaccharide capsule protected from phagocytosis, causes the pneumonia and meningitis in the adult and otitis media in the children. And if the Optochin disc is resistant, then we go for the biolusiculin test. If the biolusiculin test is negative, then it is Veridon species, which is a normal flora of upper respiratory tract and causes the dental infection. If it get into the bloodstream, it causes the subacute bacterial endocarditis. Aeruginosis group grow into the low oxygen and may be found in the abscesses of a brain and abdomen. Biolusiculin positive, PVR positive, PYR positive, NaCl 6.5% positive is Enterococcus, and biolusiculin positive and PVR and NaCl negative is group D streptococci. Further, we will go to the beta hemolytic group. In the beta hemolytic group, we have again PVR, bisitracine positive, are sensitive is pyogenes, group A, strep throat, scarlet fever, and PVR and bisitracine resistant is heparate, further differentiated on the basis of heparate, and camp, which is positive in the group B, which is a Gilecti, bile positive, normal flora of genitourinary tract, can transmit it to the babies, causing the neonatal sepsis, meningitis, pneumonia, antiphagocytic capsule containing the salicylic acid. And if the heparate and the camp is negative, the biolusiculin is again used to differentiate between the Enterococcus, which is positive, and also PVR and NaCl positive, and group D, which is positive to biolusiculin but negative to PVR and NaCl. And if the biolusiculin is also negative, heparate is also negative, camp is also negative, and beta hemolysis is there and resistant to bisitracine and PVR, then we suspect it is beta hemolytic strep, which is not group A, B, and D. So E and F.